Mechanisms of hormonal interactions. The mechanisms of hormonal interactions with the muscle tissue depend on several factors. First, when exercise acutely increases the blood concentrations of hormones, a greater probability of interaction with receptors is possible. If the physiological function to be affected is already close to the genetic maximum, i.e., with little adaptive potential left. However, the receptor is not sensitive to the increased hormonal exposure. For example, a muscle cell that has already reached its maximal size with long-term training may not be sensitive to hormonal signals from the body to stimulate further protein accretion. How and when this reduction in receptor sensitivity to hormonal increases occur in human muscle is unknown. However, genetic predisposition ultimately limits increases in size. Second, because adaptations to heavy resistance exercise typically are anabolic in nature, the recovery mechanisms involved and related to increases in the size of the cells. Third, mistakes in exercise prescription can result in a greater catabolic effect or an ineffective exercise program. As a result, hormonal mechanisms either adversely affect cellular development or minimally activate mechanisms that augment hypertrophy. The combination of many different mechanisms is thought to stimulate exercise-induced hypertrophy. However, not all force production improvements can be explained by muscle mass increases alone. Neural factors affect this process too. The integration of the nervous system and the various hormonal mechanisms is different in trained and untrained people. In addition, certain hormonal mechanisms, such as those mediated by testosterone, may not be operational in both men and women or fully operational at all ages. A wide array of hormonal mechanisms with differential effects based on program design, training level, sex, age, genetic predisposition, and adaptational potential appear to provide a myriad of possible adaptation strategies for the maintenance or the improvement of muscle size and strength. Hormonal changes in peripheral blood. Hormone concentrations can be determined from blood samples drawn from athletes at various stages of training. While interpretation of blood concentrations of hormones can be tricky, it is only one part of the whole hormonal response puzzle. Such data provide an indication of the status and or response of the glands or the functional status of the mechanisms controlled by the hormone. It should be noted that peripheral concentrations of hormones in the blood do not indicate the status of various receptor populations or the effect of a hormone within the cell. It is typically assumed, however, that large increases in hormone concentration indicate higher probability for interactions with receptors. Decreases in hormonal concentrations indicate several possible fates for the hormone, including higher uptake into the target tissue, receptors, greater degradation of the hormone, decreased secretion of the hormone, or some combination of these. Many different physiological mechanisms contribute in varying degrees to the observed changes in peripheral blood concentrations of hormone, including the following. Fluid volume shifts. Body fluid tends to shift from the blood to the cell as a result of exercise. This shift can increase hormone concentrations in the blood without any changes in secretion from the endocrine glands. It has been hypothesized that regardless of the mechanism of increase, such concentration changes increase receptor interaction probabilities. Tissue clearance rates. Tissue clearance rate is the time it takes for a hormone to go through the circulation of the tissue. Hormones circulate through various tissues and organs, with the liver being one of the most major processing organs in the body. The time delay as a hormone goes through the circulation of the liver and other tissues, e.g. lungs, keep the hormone out of the blood circulation and away from contact with target receptors in other parts of the body, or can degrade it and make it non-functional. Hormonal degradation. That is, breakdown of the hormone itself. Various pooling of blood. Blood flow back to the heart is slowed by pooling of the blood in veins. The blood is delayed in the peripheral circulation due to due to the increased muscle activity. Muscle contractions greater than 45 the maximal, thus pooling of the blood can increase the concentration of hormones in the various blood and also increase time of exposure to target tissue. Interactions with binding proteins in the blood. Hormones bind with specialized proteins in the blood that help transport it. Free hormones and bound hormones all interact differently with tissue. Ultimately, it is the free hormone that interacts with the membrane of other cellular receptors. Receptor interaction. All these mechanisms interact to produce a certain concentration of hormone in the blood, which influences the potential for interaction with the receptor's target tissue and their subsequent secondary effects, leading to the final effect of the hormone on the cell. Adaptations in the endocrine system. Although organs, such as muscle and connective tissue, are the ultimate targets for most resistance training programs, many adaptations occur with the endocrine system as well. These changes are related to the changes in the target organs and the toleration of exercise stress. The potential for adaptation in the endocrine system is great, with so many different sites and mechanisms that can be affected. Following are examples of the potential types of adaptation that are possible. Amount of synthesis and storage of hormones. Transport of hormones via binding proteins. Timing needed for the clearance of hormones through the liver and other tissues. Amount of hormonal degradation that takes place over a given period of time. How much blood to tissue fluid shifts occur with exercise stress. How tightly the hormone binds to its receptor affinity, which is an uncommon response to exercise training. How many receptors are in the tissue? The magnitude of the signal sent to the cell nucleus by the hormone receptor complex or secondary messenger. The degree of interaction with the cell nucleus, which dictates how much muscle protein to produce. Hormones are secreted in response to a need for homeostatic control of the body. The endocrine system is part of an overall strategy to bring physiological functions back into normal range. These homeostatic mechanisms controlled by the endocrine system can be activated in response to an acute, immediate, resistance exercise stress, or can be altered after a period of chronic, over longer periods of time, resistance training. The mechanism that mediates acute homeostatic change to acute resistance exercise stress typically is a sharp increase or decrease in hormonal concentrations to regulate the physiological variability, such as glucose level. A more subtle increase or decrease usually occurs in chronic resting hormonal concentrations in response to resistance training. For example, the subtle increases in testosterone over the course of a resistance training program may help to mediate changes in protein synthesis, thus leading to increased muscle fiber size. The primary anabolic hormones. The primary anabolic hormones involved in muscle tissue growth and remodeling are testosterone, growth hormone, and insulin-like growth factors, which are discussed next, as well as insulin and thyroid hormones, which are examined in greater detail in other sources.